Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. I hope you are doing well and I hope you all are fine. Dear students, we are in a continuation of the chapter that is Principle of Inheritance and Variation. In some of the previous classes, we have discussed already about the linkage, types of linkage, the chromosomal theory of inheritance, and the Morgan's experimental verification for the chromosomal theory of inheritance. That all thing we have discussed. But today, the topic which we are going to discuss in this chapter, that is linkage mapping and chromosomal mapping. So, linkage is a physical association of gene or a side-by-side presence of a gene or how far away these genes are present. What will be the effects? If genes are close apart, then the linkage is more. If genes are far away, then the linkage is less and the chances of recombination are getting increased. That all thing we have discussed in the previous class. Today, how these all genes are positioned, how they are arranged, and what is the distance between them. If we need to identify that on a particular chromosome, then we have to understand this topic, that is a linkage mapping or a chromosomal mapping. So let's understand. So first we define what is linkage map. If we talk about linkage map and we define it, then it is a straight line. It is a straight line in which genes are arranged in specific sequence separated by some distance, separated by some distance, which is proportional, to their percent crossing over. Okay, so in linkage map we are going to align all the genes in a specific sequence and they are separated by some distance. The distance will be proportional to the percent crossing over. Now what is percentage of crossing over, that we'll discuss now. But be remember this linkage mapping, or we can say the chromosomal mapping idea, and a linkage map was given by, that is Alfred Stuart. So Alfred Stuart, who was a student of Morgan, and this phenomena is given by him. What Alfred Stuart did, he used the frequency of recombination, used frequency of recombination between gene pair on the same chromosome on the same chromosome as a measure of as a measure of the distance between gene, is a measure of the distance between gene, and mapped their position, means the position of a gene on the chromosome. So here the percent crossing over or a recombination frequency is going to represent the distance between genes. Okay. So it is known that it is a type of indirect mapping because it is done based on the result of the test cross. And here the question is, what is recombination frequency or percent crossing over or a crossing over frequency, that we need to understand. So if you want to understand, then we can use a one formula for this thing. So recombination frequency or percent crossing over percent crossing over or crossing over frequency or crossover value. These many terms are used for this recombination frequency. The recombination frequency can also be called as a percent crossing over or a crossing over frequency or a crossover value. Now, this recombination frequency is equal to number of recombinant offspring or non-parental offspring upon total number of offspring multiplied by 100. So this is the actually the formula for the recombination frequency. And this recombination frequency is also known as percent crossing over or particular this one. Now, if we write 1% crossing over, then it is equal to 1 map unit distance. Or 1 centimorgan. This map unit is used in case of prokaryotes, and this centimorgan is used in eukaryotes. So 1% crossing over is equal to 1 map unit or 1 centimorgan distance is there. And one of the important thing is that recombination frequency is going to be maximum greater than or equal to less than or equal to 50%. Sorry. Recombination frequency is going to be always less than or equal to 50%. Means the maximum it is going to be only 50%. Okay. Greater than 50% recombination frequency cannot be observed. Okay. So this is the one of the important thing which you will see here. So if the particular distance, if we see between the particular gene, suppose, let's talk about some of the example. If suppose the distance between gene is distance between gene is 33 map unit, then the percent crossover value or recombination frequency is going to be 33%. If the distance between gene is 48 map unit or centimorgan, the distance is this much, then the cross recombination frequency is going to be 48%. But if the distance is 51 map unit or centimorgan, then the recombination frequency is going to be 50%. If the distance is 66 map unit or centimorgan, then the recombination frequency is going to be 50%, because the recombination frequency cannot be more than 50%. So if the distance between gene is seen above 50%, so for that the recombination frequency is going to be 50% only. Up to 50%, whatever will the distance between gene will be equivalent to that, the recombination frequency will be observed. But if the value of distance between gene is more than 50%, more than 50 map unit, then it will be equal to the recombination frequency, that is 50. Okay. In such a way you can solve this type of particular question. So now, to understand this, we need to understand one question and go through this question, so it will help us to properly understand this. So if we take a question is saying that if a distance, if a crossing over frequency, not distance, or we can say recombination frequency, if a recombination frequency between genes A and B is 20%, between gene B and C is 6%, then between A and C is 14%, and A and D is 4%, then what could be the arrangement of linked gene on a linkage map? So if we solve this type of question, if the distance between A and B is 20, recombination frequency is 20%, then we need to first identify the distance. So distance between gene, that is between A and B, is 20 centimorgan or map unit. Okay, B and B. Between B and C, it is 6 centimorgan or 6 map unit. In between A and C, it is 14 centimorgan or map unit. And between A and D, it is going to be 4 centimorgan or map unit. So if we see the distance, then you can see that if we make a straight line and we make a distance, then between A and B you will see that it is 20 centimorgan or map unit. Now then between B and C, okay, and A and C is 14% and B and C is 6%, so it is going to be 20. So between A and C, you can make it is a 14 centimorgan or map unit. And between C and B, you will see the 6 centimorgan. So it is going to be 20 total. Now between A and D, it is going to be 40%. So A and D, it is going to be 4 centimorgan this side, or between A and D, it can be 4 centimorgan this side. So the arrangement of gene on a linkage map can be D A C B or A D C B. Okay, this way the arrangement of the gene on a linkage map can be possible. Okay, so this is the particular linkage map, or we can say the arrangement of gene. Let's discuss one more question. And the question is, an organism is homozygous for A and B gene. Now it is crossed. It is crossed with pure wild type. It is crossed with pure wild type. Their F1 is test crossed. Their F1 is test crossed, and a result of test cross of test cross are as follows. Plus plus slash AB offspring, genotype carrying offspring are 956. Then AB slash AB means AABB. Here it is capital A small a, capital B small b. Here is capital plus plus represents the dominant one. So dominant A, dominant B. Here these are 944 plus B slash AB is equal to 293, and plus A plus B are 307. Okay, so now these are the results. So the question is, Then find out number one, type of inheritance, and number two, percent crossover or recombination frequency and distance between A and B gene. So if you can see here the given offsprings of the F2 generation, these are the parental offspring and these are recombinant offspring. So if recombinant offspring are present, then obviously we can say type of inheritance is going to be incomplete linkage, because the crossing over or recombinant offspring or non-parental offspring can be obtained only in case of incomplete linkage. Incomplete linkage is want to be observed. So the type of inheritance is going to be incomplete linkage. But if we want to identify a percent crossover recombination frequency, then we need to know about the recombinant offspring. So recombinant offspring are 293, okay, plus 307. So these are total going to be 600, okay. So 293 plus 307. And total if you see then 956 and 944. So these are 900, 900, 1800, 1800, and these are the total 100. So 1900, 1900, and plus this 600. So it is going to be 2500 into 100. If you identify this, then it is going to be 600 by 2500. And if you talk about 600 divided by 2500, then you will get into 100, then you will get a 24% recombination frequency. So here recombination frequency is going to be 24%. So the distance between gene is going to be 24 map unit or centimorgan. Okay. So this is the what a particularly seen in case of this type of questions. Okay. So I hope you understood this. You all are getting this. What is linkage mapping and what is chromosomal mapping? How it work? That all thing we have discussed. Okay. So till the next lecture. Thank you everyone. Keep continuing with this lecture series. You will gonna enjoy all the topic which we will discuss in the further lectures. Okay. Thank you.